Okay, I'd like to welcome everybody to the March 19th meeting of GLIMPRINT and the IMAG Working Group on multi-scale modeling and immunology. This meeting will have one talk and discussion by Reid Shabman from NIH and NIAID, talking about Data Ecosystems Initiative at NIAID. As always, I need to remind people that this meeting is live streamed and recorded and made available in edited form for future viewing, so it is a public meeting. I want to thank our organizing team. Tom Helikar, myself, Jim Sluka, Lorenzo, and Ellen are always available to take your suggestions, comments, any way we can improve things. We're glad to do it. Please feel free to reach out to us with your suggestions and ideas. We have a variety of communication channels that work better if you use them. Please help us advertise so that we have better attendance and that our resources, online resources, are made available to the community as much as possible. If you have a publication to share, we could put that up on the IMAG Wiki. Contact Jim Sluka if you want to do that, jsluka@iu.edu. There is a new version of the GLIMPRINT website. You can post announcements there, workshops, publications, grant opportunities, job openings, and so on. Again, if you want to do that and you're having issues with it, please reach out to jsluka@iu.edu. We'd be glad to have your help building that website. And I want to thank Jacob as well for his work doing that. Coming up, we have a bit of a break, and then in April 16th, the continuation of the lessons learned series, and then in May 14th, Kevin Flores is going to be talking about hybridizing data science and mechanistic models, which is a topic of great interest to this community. If you, your students, your postdocs would like to speak on March 26th or April 9th, please reach out to us, reach out to me or Lorenzo, or put that in the chat in the meeting. We always are happy to hear again from people who've spoken before. We're happy to have your students and postdocs speak. We also always are glad to have your suggestions for people who might not be modelers per se. If you have ideas of people who've done interesting work in experimental techniques, in measurement, in drug deployment, in commercialization, these are all interesting topics. We've had people speak, everything from critical care doctors to people working in regulatory agencies to people doing drug development, economists, journalists. So please think broadly about topics that would be of interest in the general area of immune response and modeling thereof, and send us your ideas. Lorenzo is very effective at getting people to say yes when they're invited to speak. I'm always calling attention to the European roadmap for virtual human twin. It's a great document. I encourage people to look at it. We still have our open VT reproducible multicellular modeling competition till the end of the month. We would love to have your submissions for that. Any multicellular modeling paper that was published doesn't have to be on immunology, published before March 31st, 2026 in the past, so it can be published in 2024, 2025, 2026. We'd love to have you submit to that. It's not a hard application, so go to openvt.org and you can apply there. We'd love to have your ideas. We will not have a steering committee meeting tomorrow. We will resume in 2 weeks discussing the organization of an annual meeting, the seminars, and also the website, and also the upcoming workshop that's being led by Jim Sluka and others. It's going to happen in Atlanta. Are there any other business announcements before we get to our main topic? Okay, so today we're very pleased to have Dr. Reid Shabman from NIH, NIAID, Acting Director of the Office of Data Science and Emerging Technologies, who's going to be talking about the data ecosystem from that perspective. He's spoken to us many times over the years. He's been a great advocate for the development of multi-scale modeling community, and we're very pleased to have him speak to us today. I hope we'll have a lively discussion afterwards.
I'm going to try to go through this pretty quickly. The slides can be available, but I think folks probably want to get to some discussion. It's an opportunity to show that we're a small office, but we're really hear what you all are doing and trying to do what we can as a group of 7 or 8 to advance some things moving forward. And so I'm going to just sort of touch on some vignettes that our office works on very, very hard. And just we've called this recent advances across the NIAID data ecosystem and like what are our perspectives on it. And always looking forward to hearing like what can we do better, that type of thing. And our goal really being small is really not to create everything ourselves with a partner. So to jump into this, the agenda right now is going to be a quick update on NIAID. I think most people are familiar with the NIH structure. We're one of 27 institutes, centers, or offices. We have new leadership, so I'll talk about that. And then some vignettes of our office. And then I want to get to some representative NIH updates as it might apply to what you're thinking about for funding and other things that you've heard in the news. And again, I'm middle management, so I don't have all the answers. All right, so we have a new acting director, Jeffrey Taubenberger. He is coming from the intramural division at NIAID. He's got over 300 publications to his name. Very smart, thought-provoking individual. Claim to fame really was sort of resurrecting the genomic sequence of 1918 influenza virus. One of the things that he co-published with Dr. Bhattacharya and John Powers, who is our deputy director, is this new vision from the National Institutes of Allergy and Infectious from NIAID, where they really are talking about 2 priorities. And I would just point you to this paper if you want to learn more about what's in the paper, but really just to bring it to the top. We want to address the most impactful infectious diseases. That's not necessarily any different than what we had been doing, but also to support innovative research that addresses fundamental studies in immunology, allergy, and autoimmune diseases to improve patient outcomes. And so really just sort of reiterating that there's 2 sides of the coin at NIAID. One is the infectious disease and the other is allergic. Also, how that relates to chronic disease and whatnot. So there's a little bit more there. I'd point you to that paper. So who are we? We're in the office. We're in this ODSET office, and since I'm from the government, I will show kind of an org chart here. We're the second biggest IC. OD stands for Office of the Director. I'm not going to go through all the acronyms. What you need to know is these are the funding divisions: DMID, DAIDS. These are the intramural divisions: DIR, VRC, DCR. DEA is kind of what coordinates all of this funding, and we sit in the Office of the Director. So our job. Our office was created in 2019, so we're a small office with the vision to lead NIAID's data science strategy to ensure the effective use of data and computational methods across its research and training programs. And we've done a lot of work in this space. We kind of think about data science. If you think about NIAID, we issue about 6,000 notices of awards a year. That's a little bit misleading because if you have an R01, we're counting that every year, right, when you get your type 5. From that, we've started to say, okay, within those 6,000, if we make a cutoff from computation, if we throw out the experimentals, yes, maybe they're using AI experimentally, but if the majority of the project is computational, let's use that threshold. And then let's sort of bin those computational projects into different categories. And that's what we've been working, and Sydney's on the call here. We've been working very hard on this effort, where we basically have categories like artificial intelligence, career workforce development, computational biology and systems modeling, DRKBs, as we call them, data management centers, and software engineering. I'm not going to go into kind of the logic behind all this, but just know that this is how we're thinking about it. So that we can start to identify gaps and trends that are going. So like one trend is that artificial intelligence grants, you see a spike in that, obviously, right? And that almost corresponds with the large language models becoming commercially available. You can almost see an inflection point in the number of awards. And so if I had to pick one, just as a point is and this is an old paper, but I like to use this example. So out of the 6,000, what's one that I could point to? And I'm going to get to some others that you might see later, is that AI ML on large data sets can support decisions in the ICU. And the reason I picked this is because you're kind of bringing in emerging technology where you basically are using machine learning to take a lot of data that comes in from ICU patients. So basically, two people come into the ICU, they're the same age, or the same gender, they both have the same disease. One lives, one dies. What happened? And so can we start to use machine learning or advanced technologies to take the data, not just the patient EHR records, but also the multiomics data, and put all that together? I think like things like N3C do a lot of that, but they didn't ever really bring in the multiomic data. And can you start to come up with like favorable states, intermediate states, unfavorable states, to kind of like say, hey, this person's about to go to a less favorable trajectory. Maybe we should do something. And can we improve this sort of survives, dies outcome? So that's kind of like obviously it's easy for me to say that as a program officer, not easy in the real world. The reason I'm using this is because this data is available. It's in things that we manage here, like dbGaP, Access Clinical Data at NIAID, SRA, and you can get it from Northwestern. And this data has been used for codeathons. It's published in 2023, but they continue to collect this data and sort of build this foundation out. And so I just think it's like a nice way to sort of show the clinical connection with data availability and data sharing. But we could spend hours going through different examples. And so, but what our office really tries to do, I pointed to that paper a couple minutes ago, where the NIAID strategic aims currently are infectious diseases as well as immunology and allergic and autoimmune diseases. You can then say, well, underlying those things are technical priorities. So these are just a couple of technical priorities that we've identified from the Taubenberger paper, from NIH town halls, from the gold standard science, and those are NAMs, emerging technologies like AI, the importance of reproducibility, the importance of data sharing, and probably the most important, workforce development in the data science arena, right? And our activities are really kind of designed to align to these technical priorities. And so if you look at it one other way, we kind of have these four pillars. So everything I'm going to talk to you about today kind of maps back to these four pillars in our office. We think about data infrastructure, developing an interoperable network of data and analytical tools to promote data sharing. We think about research programs. How can we develop research programs to improve the use of data science methods and approaches? We think about training programs and workforce development. What are the training programs we can use? And we think about governance. How do we improve the policy and framework for equitable sharing and computational methods? And we think about this, that data feeds into software, feeds into models. So it kind of connects back to this group here. And what I'm going to do is try to show a vignette of each one of these. So we'll start with them. So it'll map back to these four pillars. So going into infrastructure, like this is our vision that we're working on. It's imperfect, but this is where we're trying to go, is what if we had a NIAID network? And I'm speaking from the NIAID perspective. It can go off the rails pretty quick, right? Well, what about NIH? What about the world? We're five people, so we focus on NIAID, and hopefully the methods and approaches that we're using are scalable, extendable, et cetera. But a NIAID network of interoperable data, analytical tools, and services is really where we'd like to go. And we're trying to do this kind of through two ways. And I'll get back to this. I kind of took this slide from another presentation, so it's not flowing exactly right because it's talking about AI. But you can think up here is we have a search portal. And which I will talk about in the next couple slides, that allows you to find data from all these different data sets, biospecimens. Can we find computational tools and models? Can we find NIAID clinical data, clinical trial data? Can we start to be interoperable across other data ecosystems? And that's fine, but can we take it to the next level, and can we put like AI on this? So now we're starting to move to a spot where this is in staging, but we can start to have conversational search. So instead of Elasticsearch, we can start to ask more conversational questions sort of based on the JSON metadata that we have for over 4 million records. And so this is kind of where we're going, and I'm going to talk about that on the next couple of slides here. And so the immediate challenge here is, how do you harmonize metadata across the landscape? So on the left here is if you think about all the data we're thinking about, it goes all the way from like immune epitope database, IEDB, all the way up to like the MaxWise combined cohort study, which is HIV longitudinal clinical work. And you can imagine that there's a lot of different types everywhere in between. And so I think I don't know if John, you might have been around at the time, but we a couple years ago, a paper was put out kind of like, how do you address these barriers? I was involved in that. And also Laura Hughes, who's now at Genentech, she was really leading this effort with Ginger Sung at Scripps for developing a standardized but extendable framework to the findability of infectious disease data sets. And this was they agreed upon this metadata schema that would allow us to connect all these different types, because we said, hey, there's some fundamental elements here that are common, basically. And if we get that to a point where we can map all these resources back to this schema, we can create a search portal. And so this is our solution right now, is this NIAID Data Ecosystem Discovery Portal. Our office manages this through a contract. It's a centralized hub for data sets and computational tools. You can also, we index biotools, so it's not all tools, but basically you can discover immune-mediated infectious disease tools here from about 4 million resources. This 80 is a little bit, it's about 45, but we have if you go to the landing page, we have about 80 different repositories listed. 30, like we'll say 35 of those, we're not able to index at the data set or sample type level yet, but if you type in HIV repositories will come up even if we can't get to them. So so that's kind of our technical challenge. We're trying to track research and funding programs as well, and this supports data sharing through the AI-rich metadata that I mentioned earlier. I think everybody's probably heard about this before, so I'll just say, it's kind of this funnel in all these different resources to get to a query and then funnel out into the portal. We did, if I'm not going to really go into the technical specs here, there was a paper in M Systems that just came out at the end of the, on New Year's Eve, which was funny. So I guess in 2025, that describes sort of how we harvest, harmonize, store, expose, and access these metadata. And what we're trying to do, NLM has a data set catalog they're building, cancer research, NCI has one, it's a common idea. And I don't want to really get into that right now. But I want to say that what we're trying to do is really make it better for the user to find data to support hypothesis generation and secondary use. So you can search by topic, health condition, ontology. All these metadata fields we've enriched upon, you can start to search. You can Google yourself, ask how many data, if you type in Dr. Laubenbacher, you see how many data sets are associated back with the author. So it's kind of like a PubMed for data. We have about 50 diseases and conditions you can pre-filter on. So if you're interested in data sets for one of the many pathogens we focus on, you can just go right to one of those disease pages. And if you're interested in what data is generated by NIAID programs, we index that. The caveat there is, you need the metadata, you need the funding ID active in the that we can access. So we're sort of collecting that by funding ID. So that's a nice way to, track back, where the tax dollars are going to these programs for the data. Programs will come and go. The data theoretically should live forever and be used, in the future. I'm not going to do a demo. This was for a different presentation, but our ask is, please use this portal, and we'd like to hear your. One of the questions we always have is like, well, what's the point? Like, what are the use cases? And I can come up with my use cases, like, give me some transcriptomic data sets for a clinical trial for malaria in South America. But like, that's just my bias. So like, what would be, what is this not getting for you? And we're always welcoming that. I think there's ways to contact folks there. So that's the findability piece of it. Expanding the ecosystem. We work with a lot of different repositories to advance reproducibility. So what I'm showing here are just 12, depending on how much you want to call them, they're not all like harboring data per se. So, for example, these bioinformatics resource centers are pulling data from GenBank and the protein databases at NLM and sort of curating them, right? But they still harbor some. But we work across our divisions, the big ones like Import, you've probably heard of, ITN Trial Share, IEDB, TB Portals, Access Clinical Data, the HIV resources. We've engaged with them to provide recommendations. It comes back to that graphic I showed earlier about connecting data, connecting the actual data to sort of lower barriers for data access and reuse and accelerate the treatment. So what do I mean by that? We put out this blueprint to support data sharing and reuse. We call it the NIAID Blueprint for Digital Objects in the NIAID Data Ecosystem. It's pretty straightforward. There's nothing controversial here. And quite frankly, a lot of people have said this is like not transformational, and that's fine. We see some resources as sort of gold standard high level and others that maybe are a little bit more less mature. But the way to think about it is like if you have a data resource or you're a data generator, what are the things you should be thinking about? So what are the metadata you should be capturing when you're developing an experiment, and then how do you uh, represent that metadata? Which PIDs, which ontologies, which GUPRIs are you using to represent your pathogen or your spatial temporal category, whatever it is? And if you start to think about persistent identifiers in your metadata, that's going to help. And you put that into Zenodo or wherever you put it, a domain-specific resource, it's going to help you one, get cited. People are gonna be able to find that data, use that data, cite that data. We talk about, at the ultimate end of the day, how do you get credit for that data set you've generated? Or I haven't even talked about I haven't used the word model yet, but we can get to that. And then, that's going to help us feed these data catalogs and also get to the community. So you can read this blueprint. It's on Zenodo. We put this out with a group called Go Fair US. So we're not naive to I didn't put NLM's catalog on here. We're not naive to the fact that they're beyond NIAID, there are NIHICs and others optimizing their data landscape. We have the portal, there's the Common Fund Data Ecosystem, there's NHLBI, there's NCI. Obviously, this is $1 billion investment across industry. And so what we're really thinking about is, just kind of come up with methods and approaches, sort of a grassroots. We're thinking bottom up from our space so that if we have kind of connectivity across our resources, well, maybe that's generalizable to others, and maybe that's of interest to the bigger fish. So these are the acknowledgements. The portal is done by the Scripps team, and that sort of blueprint I mentioned on the previous slide is Christine Kirkpatrick and her team at Go Fair US. This is supported by a contract at the bottom here. So it's a large group of folks that are working on it. I just happen to be speaking to it. So I'm going to pivot now to that second pillar, which is research programs. And this is really to develop programs to improve the use of data science methods and approaches. We have a working group here at NIAID that we develop these pillars with them. So if you think about data to software to models, I don't know if that's the perfect analogy, but now we're kind of moving into the modeling space. We've seen a lot of challenges in NIAID in the infectious and immune-mediated disease, or IID, modeling landscape. One of those challenges is model and data sharing. How do you talk about sharing data? How do you share models? How do you reproduce models? We have a large dispersed modeling community. We have NIAID programs, have the MIDAS network. We have IMAG. We have, this might be outdated, but we have these other modeling groups. And then a third challenge is, well, how do you make multi-scale models? How do you bridge models across biological scales? And I'm biased. I'm at NIAID, so I'm talking biology. So that's kind of our focus. And so you sort of have these things to think about. We have divisions here, that are data generators. I could give you a list of 50 programs for each of the things in the division of AIDS, but there's structural biology. There's the HIPSI program. These are big data generating centers within the immune space of the world. You have the BVBRCs that are sort of capturing all this microbial data. And so with all those challenges that I've sort of mentioned, one of the things that we've been working on for the last couple of years that I think most on this call are familiar with is this center for, we were able to put out a notice of funding opportunity for the Center of Excellence for Systems Modeling of Infection and Immunity across biological scales, sort of building the plane as we fly it. But the idea was can we create something that's kind of dual purpose? One is for, advancing multi-scale models, but also to connect the community. This is a large group of folks that contribute to this from these different divisions, which is somewhat unique. Usually our divisions just do our division's interests, but this is coordinated by us with project scientists across. And Dr. Glazier was kind enough to come give us a talk at our meeting in January. This is at Duke University. They have a website, immunescale.org, and I'm just going to give just a quick highlight of who they are. So Clyburn Chan and Keith Reeves are the contact PIs here, and we have, this is this multi-project type of mechanism. So we have 3 research projects, and we have a community outreach core and a model and data sharing core. And I really think that for this group, this is going to be most interesting. I mean, the science is interesting, but I think for takeaway would be in a couple slides here. And it's a multidisciplinary team, which I won't go into because I'm not the expert, but really they kind of had two visions here, which was innovation. How do you go from, siloed individual models from the molecular to the population level to multi-scale models, and then how do you like organize those models, right? So it's easy to make a picture, hard to do. And then so that's kind of like the research project, but then like how do we like build a community? And we think of concentric circles. So like I was like thinking, well, we start with NIAID, but then how do we get back out to NIH, and then how do we get to USG? That type of thing. But like it's only been less than a year, so this is building the plane as we fly it. And I'll sort of pitch that if you go to that immunescale.org, you can actually sign up to join and be a member. And we're working with them on what that means. Will they contact you? We want to connect with this group, at some upcoming meetings, because what I've learned from this group is that they're really, really excellent at what they're doing. They're still very interested in expanding their connections beyond the research triangle, which is where they're located. So this is really technically heavy, but this is just, to say, if I asked GLIMPRINT for a slide that described the projects, and this is what I got. And so the point here is that, these projects are across scales. So it goes from antibody to repertoire is kind of the scale, where you're kind of modeling antibody protein interactions. Research project 2 gets a little bit bigger. You're getting to like NAMs, organ on a chip, modeling cells to lymph nodes, and and usually the perturbation here is a virus. It's HIV, herpes, respiratory viruses, and then research project 3 starts to get to modeling the individual to population level to sort of connect how individual infection determines spread. And this is just to show that they are able to do this, and they have protein language models for RP1, reduced order models for project 2, and stochastic epidemiological models in project 3. And I'm not going to try to explain what they're doing, just to say that you should email GLIMPRINT if you're interested. But I think more interesting to this group is the model and data sharing core. And I'll talk up and so how do we connect the data and models that you're building across the community? So the way they made this slide is not perfect, but the way I see it is there are the MISM researchers. We're trying to sort of connect the data and potentially models in our portal. We have biomodels, we have Midas Network and others. And really what they would like to hear about from you all is like, what repositories and platforms do you use to share data and models, and how are these repositories structured? Really get into the technical details of FAIR and CURE principles with them. And I'm always happy to be a point of contact there. As we sort of move this project forward, they're basically in year 1. Our vision is that we get more folks involved, at least aware. They have webinars, you can come present there. They're going to have meetings. What are ways that we can start to build the community to come up with consensus. So it's not to just build yet another program. It really was sort of from the perspective of NIAID has 6,000 awards. A lot of those are computational. We got to organize those. We realize that the modeling methods and approaches beyond NIAID are going to be very important as well. And going like, and if it's all put together one day, we get these multi-scale models together, we can start to get closer to some of these some of these slides, right? So on the left here is a slide that Reinhardt, James, and others are very familiar with. I like to use it because it very clearly illustrates the concept of how you could take different biological data, put it together into some sort of in silico AI-enabled model, which we're calling a digital twin here, to help drive personalized care. And so I think, 5 years ago, I think there was a lot of skepticism here. I think that the world has changed with AI. I think I think this is becoming a very popular topic now. And there are papers that are coming out. I just gave one here about virtual twin for RA. And this is powered by Import Data, which is a NIAID funded resource. And I think that Import is one of those that we're working with that really is having a lot of this AI ready data that can feed into some of these things. So it's more than just Import, but I wanted to say that, Import is really kind of supporting both of these ideas of both immune digital twins and specific use cases within that space. Our office also works on training programs and workforce development. Because we're a small office, We also were relying on this large center to help with that. Training is a priority of the MISM community, so they have this community development education core, and this is ongoing. They basically came up with some ideas for how to like connect to the community. So they have started with a 10-week summer internship, and this is all kind of in the past. But if you keep in mind, like, if all goes well, this will kind of cycle again, right? This summer they're working on a summer internship at Duke University. They're working on sort of what part of the NOFO prescribed pilot awards for small projects, and the response was overwhelming. And they're working on sort of through those logistics here in year one, and we're supporting them in that space. And then they're also doing ongoing listening tours to identify areas of collaboration. So I put the link here to connect with Gina Maria and Justin when you're talking about those types of activities and how it relates back to the center. I think they would be open to hear what you're thinking about. This is just a point back to the fact that our office makes strategic investments in things like R25 awards. We've tried to add, that's sort of highlighted here. I was lucky enough to go down to South Carolina and present about a month ago. So I just wanted to highlight that this big health science community scholar program, or the C Scholar program, but we have done others. And I would say, stay tuned for updates in this space about R25s from NIH. We have also expanded data science training slots in T32s, where we tried to add additional data science training to people that had institutions that had T32s already. So we did that last year in fiscal year 25 to these 3 centers. And we also have at NIAID, we in our office, we support a fellowship program where basically it's kind of a reverse situation where people can come to NIAID to rotate around the institute and learn about NIAID. We're looking for recent graduates in biomedical informatics, computational biology, computer science, et cetera. They come here, they do rotations. There's different trainings, there's rotation hosts, and it's sort of a career development path if you want something outside academia. Sydney's on the call. She was a fellow. Meg is the program officer for that U54 I mentioned. She was a fellow. Lisa is the one behind our blueprint. She was a fellow. So they're all in our office, right? So it's pretty nice. I'll drop the link in the chat. We're accepting applications right now. The final pillar here is I'm going to try to end by about 5 minutes here. Is data governance. This is really where Sydney does her work. You can think of this cycle as researchers sharing their data, goes into repositories, and then there's secondary data use. And there's policies around how we should share our data. And then there's policies around how we should access our data. All you really need to know here is that, NIAID manages dbGaP access requests as it relates to NIAID specific data. And we also have a controlled access data repository called Access Clinical Data at NIAID, which we highly encourage you to submit any clinical data that you have into this resource. And then we also have something called TB portals, which is a nice resource for accessing tuberculosis data. So this is more on the policy side. I'm going to just say that, people always, the most common question on this side is always, well, where should I put my data? You say I have to put it in a repository. Where should I put it? And if you really read the fine print, they're like, don't put it in a generalist if you don't have to. Put it in a domain specific. So then someone's like, well, how do I figure that out? So it's kind of complicated. So one thing our office did is we published this paper where we basically looked at 300 different data resources and made a series of assessments of these resources and sort of whittled it down. And the punchline here is we got to about 58 of these 300 are domain specific. And so like, I'm not counting like geo in this, right? So this is like more like IID specific. And 19 of those are actually supporting data deposition. So we kind of went from about 300 to 19, but we have information about all. And then there's a sort of a questionnaire that we developed to assess these 19. And there's a lot of information about these resources. So it's a nice document for you to reference. And we encourage you to go check it out, because it may help with like model development and things like that nature. So everything I sort of breeze through here can be found on our website. We have a NIAID Data Science website. It's constantly evolving, so we haven't been updating it as much recently, but you can basically everything I talked about you can find here. And we do try to like, if you have public events, we can promote those on our events page. If you're curious about the policy stuff I talked about, we have a lot of stuff about the policy you can go read. And we've tried to like synthesize the NIH policy around data sharing to something that's like more useful to the NIAID community, or maybe just to anybody that doesn't have a lot of time to read that website, that type of thing. So I do encourage you to go check this out. We highlight publications. We're looking for publications that come out that use NIAID funding. So if you have in the data science space, if you have one, happy to put it there. I don't know what the traffic is on this site, but it's, it's something. This is our team. We're pretty small, great team, hardworking team. Everybody's always putting in 120%. We have sort of this data science research and training initiatives arm, and we have this data sharing policy guidance arm. And we have a lot of consultants. Wilbert was the director. He had to retire last year for health reasons, but remains very engaged. So I just want to go bigger, go to representative NIH updates. I got about 4 slides here. I would encourage you to just bookmark this page. Everything is about centralization right now, so you can track NIH policy changes through this centralized website. So if you have questions about how to, submit awards, grants, everything like that, how to submit grants, questions about awards, payments, communications, it's all on this website. Things that I think you already probably know. There's this push to prioritize human-based research. NIH will no longer issue funding opportunities for exclusively on animal models of human disease. How do we prioritize the development of NAMs? And Nicole Kleinschruder is sort of pushed that very successfully, right? And I think that's not lost on this group. You can still use animals, but I think it's more like, how do we reduce the number of animals? And there's a lot of policies on that. Notice the funding opportunities, like what I put out, what I mentioned about that U54, like that's a notice of funding opportunity, which is different than you submitting an R01. The notice of funding opportunity basically says, hey, the government is interested in this specific area, so let's write a proposal to address that specific area. Whereas investigator initiated R01 would be like, I have an idea, and in both situations it goes to peer review. In the investigator initiated situation, it goes to peer review, and it's kind of like a mishmash, right? Kind of within that general domain area. And then with the NOFOs, we would typically it goes into like what's called a special emphasis panel, where we're like getting expertise around that specific idea. But the pendulum has swung back, where we've gone from like 800 active NOFOs, as of 1/1/25 to many less. It's not that we're getting rid of them; it's just to empower investigator-driven research. With that non-animal model idea in mind, I think is a good combination. And to that end, like, so what do you write about? There's this thing called highlighted topics, and highlighted topics is this thing called NOCES has gone away. We now have highlighted topics. Highlighted topics are centralized areas of interest for NIH. So you go to this website and it'll say, hey, NIAID is interested in research on short-lived plasma and long-lived plasma cells in humans. And you'll click on this link and it's going to tell you in about 3,000 characters why we're interested in that. And then you can submit your grants on that. And so I would say bookmark this link, keep an eye on it. What I can say, I can't really talk about anything coming out, but what I can say is that from my vantage point, there is a lot coming that will be of interest to this community with respect to data science, with respect to computational modeling, with respect to AI. You can filter by IC, and these are not funding opportunities, but they are signals to things that the NIH is interested in. So I would say, like, I would use this as kind of like your North Star for the next year. I just think that people aren't really paying attention to this site. It's not getting out to the community. And I really would emphasize folks pay attention to that. So thank you very much. I think I got 15 minutes left. Appreciate the opportunity to give such a long presentation.
Okay, thank you very much. The floor is open for questions.
Hi, Reed. Thanks so much for this overview. This is super helpful to make things more clear, given how fast things are changing and how significantly they have changed since the last administration. So this is super helpful. Thank you. A question for you on AI, and this is not specific to NIAID. This is all not just NIH, NSF, all granting agencies. So when I submitted one of my proposals, and this was to NSF in September or August, it didn't get reviewed until like March, basically. And you put in AI in September on what you're going to do, but by March, when the reviewers are
I see. Yeah.
reviewing it, the landscape, the technology has changed so that it looks like I'm writing that I wrote a grad from the prehistoric ages, in terms of what I was going to implement. How do we address this? Because everything's moving so fast right now.
So I hear you. I think it's a common problem. It's going to be very hard for me to speak to NSF. I work with NSF through IMAG, but I have a blind spot with how their review selection process works.
Yeah.
So I'm going to answer that question from the NIH perspective. You're facing a tough situation. The fact of the matter is that funds are available, but the mechanism to release those funds is inconsistent. On top of that, you have a lot of turnover on what I would call, not like folks like me as much that you talk to, but on the administrative side. Like once that score comes, like how do you actually get that paycheck at your university? There's a lot of stuff behind the scenes there. And those aren't glamorous jobs necessarily. And so once you start putting pressure on that process, you're going to start to see delays like you're talking about. And then you're going to compound that with bottlenecks with the ability to appropriate. So it's sort of like the structural moving things through the bloodstream delay combined with, well, we need additional approvals, right? So now you've kind of got two things working. So then when you use it in emerging technology, you're getting a huge delay. So that's kind of the situation you're describing. I don't have a great answer, except that you can take some comfort in the fact that you're not alone. And that everybody is dealing with that. I think it's not like yours is being held up and others are going through.
Yeah.
I think you gotta always, I think the key is like trying to stay connected to the program at NSF or to NIH and try to understand not what you can do, but giving yourself the most shots on goal. So I always tell people, like, as soon as you submit something, like assume you didn't get it and go submit something else. And if you do that enough cycles, you've kind of got a situation here where you're going to wake up one day and check your ERA comments and you're going to get something funded. But that's like a 4-year plan, right? And I know you don't have time if you're trying to get tenure or whatever it is, like to do that 4-year plan. But like in theory, that's the best way to do it, in my opinion, from what I've seen. So I think to your question, it's really just always be. I hate to say this, but it's to always be submitting and to keep in touch with your PO. I think with the emerging technology, there will be some recognition. I mean, that's going to be interesting if it comes back on these review statements that this is outdated. And I think that's now where you as the community, maybe there are always requests for information. There are ways to communicate that. I think that needs to be brought up. I mean, I think AI is moving so fast, everybody's like, what do we do with AI? Like, I don't even know what that sentence means, right? But I think
because it means something different yesterday than it does today. Yeah.
Right. And there's scientific AI and there's sort of operational AI for your email. That's not a neural network, so like, but like, so I think you've got to communicate that from your, like, that's what you can do is you can say like, look, this is affecting the ability of a review due to these delays. And I think you can make that point professionally and I think it will be heard. But that's and from our side, we can try to do the same thing. Specific examples always help, right? Like I think if you talk in generalities, it's a little bit harder. I think it's easier to have that conversation when you can be like, this grant was submitted on this day, and now look here, it's this far out, and this technology shifted. I think that crystallizes it for people that are thinking about this at the macro level.
Yeah. Okay. Thank you so much.
Yeah.
Okay. Other questions? Go ahead.
So I had a bunch of questions. Some of them are related to governance of data and models. There's a big legal problem, but one more acute. There was an educational aspect there. There's a slide about education. is the NIH offering a free vibe coding course to researchers? Because this seems to be something necessary. I had researchers ask me to show them how I vibe code. So this is something that the NIH should do to everyone.
It's a great question. I think there are courses that are available in our learning centers, but I'm not sure if those are open to the general public. Usually the way the model works is funding goes to a specific institution to develop training courses. I'm not saying this is my, and in those courses, those courses would become available. And then the question is, are those courses available to the general public or just to those universities or communities? I think it's a great point. I think the question is like, when we say training and training the next generation of scientists, I think most of the time it's interpreted as folks at universities receiving training at those universities, as opposed to the NIH providing open access courses to train the general community. I'm not saying that's right or wrong. I'm just saying, like, from my perspective, that's typically how it's happened. But I'll take that back, Jacob, and look into it, because I do get emails all the time about classes in R and Python, and I never actually checked to see if those are available to everybody or not, honestly. So I'll take a look. Thank you.
Jacob, did you have a question about copyright issues? He asked, when you're making your data sets available, how do you handle data ownership, copyright, and are there efforts to make public domain data sets clearly public domain so that there aren't licensing issues when those data sets are used?
So yeah, so the short answer there is is that within that, I should probably drop some links in the chat, but we do have this blueprint that talks about sort of choosing a license. Usually you're kind of if you go to something like a Figshare, right, you kind of choose your license, right? You kind of choose like what license you want for your data. Our position is, when possible, we want data to be open and open access. Even if it's got human subjects associated, I'll go quick here, but even if it's got human subjects associated with it, has to go to controlled access, that's an exception. If it's international data that's generated, we're not going to supersede any government or tribal laws outside the United States. But if this is data generated in the United States and there's no PHI, like there should be we encourage folks to make that open. I think the point you're getting back to, though, is like, how do we make these things more citable? How do we make them more findable so that people will get credit for the data that's out there? And I think the idea of like a gold standard public AI ready data set is great. Is that something, and that's the things that we think about all the time.
Reinhard, go ahead.
Yeah, thank you, Reed. This was great. It was very helpful. One of the things you mentioned that you're interested in is of course reproducibility, fair standards, and that's of course in line with sort of what everybody else says they're interested in. In my own experience, when it comes to sort of when the rubber meets the road,
Yep.
It's not so clear how invested people really are in that. So my question to you is, do you have any concrete ideas that you think NIAID can put forward to make that a more important issue?
I do. So from my perspective, it's a great, you said it exactly right. So it's a great buzzword that we're interested in reproducibility. Here's my perspective, is that I believe that secondary data use is really popular conceptually, but in the real world, it's hard. The reproducibility is hard. So if I go try to take some data and use it to make a model, it's imperfect and it's missing something, and you don't have a lot of time. So it's easier to collaborate with somebody or do it yourself to generate new data. So I think that this concept of secondary data could be, we could invest better to ensure that like this data is actually useful. That's piece one. I think reproducibility means different things to different folks. So I've always thought that like reproducibility kind of was the scientific method. So when you publish a science paper that says, arsenic instead of carbon is why this bacteria is living, that should sort itself out reproducibly in research labs around the world, right? Yes or no? I think the way it's being talked about right now is different. And then the question is that to reproduce a figure in a paper? Is that to reproduce a dogma about some biological phenomenon? Like what level of reproducibility are we talking about? My specific example for you that I'm working on, although very slowly, is, we would like, when our clinical trials are published in New England Journal of Medicine, we would like the data to be available to the public so that figures can be reproduced from these PDFs in NEJM, and primary and secondary endpoints, the data should be available for that. And so that's kind of like my quick answer to you there is, can we reproduce clinical trial data that we're publishing where we're the IND sponsor? Can you access that data and reproduce the figure? And I don't know what the higher levels, I don't know what the Office of Reproducibility is going to focus on. I'm sure they'll put something out. I'm not connected to that office, but that's kind of, in my world, that's my specific example.
Yeah, thank you. And of course, everything you said about data, I think applies to models the same way.
I think so. And I think that's kind of where I'd be interested to hear you and this team's thought, and in this MISM group is how do you share models? So I think what the MISM group has kind of brought forward is, you have them, can we recommend specific data to put into models, and can we recommend specific models for people that have data? And then how do we, and I know there's been papers about 10 credible practices for sharing models and all those types of things. And from my perspective, just people have short memories. And so how do you, without reinventing the wheel, kind of re-amplify those pieces of work, maybe through new work, maybe through this modeling consortium, there's a way to talk about model sharing, because I think that's not really captured in the data management sharing policy of 2023.
I want to echo what you were just saying, which is that making the raw data sets available that were used to generate results in a paper is not the same as making the whole workflow for generating the images that were produced in the paper available. Now,
right,
people like Herbert Sauro have defined standards like SetML that try to do that. But in fact, one uses lots and lots of different methodologies to crunch data into something that looks like an image in a paper. And those aren't necessarily workflows that are distributed and easy to publish. And so best practice in publication is a whole separate question related to, but not the same as, making models reusable or interconnectable and so on. And so then it's a lot of work to add that level of documentation to every paper for every figure is a lot of effort on the part of the community, and we don't really have good tools. Perhaps AI now will make it easier, but we historically have not had good tools to support the publication of the workflows for actually generating figures from data.
Yeah, I think your point. Yeah, I mean, if you try to make everything reproducible and shareable with no money, or resources, that's impossible. Trying to find the right balance. And I think Herbert's example you provided makes a lot of sense. I think, to me, the way I think, and Sydney's on the call and she's our policy expert, to me, it's what's justifiable, what's defendable and within your bandwidth to get towards. So that you can say, you're not going to get every experiment I ever did, but the key figure here, this is what you need. And maybe you still can't reproduce it, but this is the person to contact to get all the pieces, right? But some sort of README file to get you to that place, right? And then I think it's a I don't want to say generational, it's a cultural change. If we went into study sections and that was the benchmark and not how many papers you had in high impact journals, or if it was seen as, or if it goes into the bio sketch, I guarantee you this would transformationally change, right? Then everybody would be well, I have to make this reproducible. I have to figure out a way to communicate this README so that we can do it. And so to me, that's from everything I've heard, like that, can't think of a better way besides putting that topic on par with publications as street cred for scientists. But that's a policy issue.
Okay, other questions? Sorry, Reinhard, did you have more that you want to add to that in terms of model sharing, for example?
No, not really. I think the last point you made, Reed, is important. There needs to be an incentive. And
Right.
in my mind, I think where the pressure really needs to come is from journals.
Yep.
That the journals have to implement certain standards. I remember when I was the editor of the Bulletin for Math Biology, and I would ask people to do certain things toward reproducibility, they would just go somewhere else. You know, but yeah,
I think
without
that's
incentives, we won't get there.
Yeah, and I think that's kind of what we see we get a lot of like urgent requests for DB gap submissions because the data has to go into DB gaps or else it can go, and they need that for the submission. So if the journal, and that's kind of where our bandwidth stops. So like, yeah, final thought is if the journals started to sort of push sort of guidelines for what's required for reproducibility, that's probably going to have a more weight than anything else. I agree. That's a great idea.
I mean, the comment that Tom put in the chat about what one of his reviewers said, I think is a case in point.
Yes, yes. We're not adding any innovation. Right, right. So it's, I would say that FAIR, the acronym, from what I've seen, can kind of have some, I don't know, everybody reacts to the acronym differently. And so you have to be, you never know when I try to avoid the acronym FAIR right now. I try to speak to what it is. FAIR is not a spec that there's no defined spec of FAIR. You'll know, it's like you'll know FAIR when you see it. So I try to say like, can we make our data connected? Can we access our data? Can we use it for reproduce? But I don't say like FAIR, let's check the boxes. So I think another thing to think about there is like, how do you speak to what FAIR is without saying our data is FAIR? Because I think that can really it hits people different ways, especially in review.
Of course, model reproducibility, simulation reproducibility is a whole level of additional complexity over pure data reproducibility, which is already a very hard problem.
I think that's kind of the reason that no one's talking about it, and that's the opportunity here. If we're going to have digital twins or multi-scale models, there's this sort of technical spec that is underlined that I think is kind of not really getting the focus. Everybody wants to talk about the picture of the immune digital twin, but not talk about how are we going to connect these things. I mean, I'm sure the scientists do, but not, but you know what I mean? So like, I think there has to be some sort of spec there that's an opportunity.
I think there's always a bit of a tension between the scientist who wants to be able to do, quote, whatever I want, close quote, and the need for standardization and to some extent imposed architectures, which are needed if you're going to allow that kind of interoperability and shareability. And of course, we accept that in all sorts of situations. We all use the same operating systems. We use the same editors. We use the same PowerPoint or slide presenters. And so in those cases, we don't necessarily feel that we're unduly restricted. And those are extensible systems in their own right. But we don't have that level of sophistication in the area of computational modeling that we do in these other contexts. As you were pointing out at the very beginning, you have these metadata annotations for your data sets. And I'm sure that agreeing on those was not a trivial thing to begin with. And with access for computational models, there are additional information that need to be exposed to make them usable. And I'm not sure that there's a general agreement on what those are. People like Iran Agman with Vivarium are trying to develop frameworks for doing that. There are European efforts in that area, but they're still not anywhere near as far along in terms of the development as we are for, in a sense, static data sets, you might say.
Yeah. I mean, 2 quick points on that. One is, I think that's kind of why our office exists, is strategic partnership with folks to do those things that aren't glamorous, that we're talking about. But then we're small. So then the question is, well, who else can I talk to? And I think the Office of Data Science Strategy is good there. Like there are a small number of us like Grace and friends that exist across the different ICs that are interested in what we're talking about, that are not the disease program officers, right? And so I think Office of Data Science Strategy is always a good place to like go with this, because these are the types of things they're thinking about. And then with respect to connecting and schemas and different things like that, like we're facing this right now. So I think it's all about, and this is from this is from someone that knows enough to be dangerous. So I'm not a technical expert, but it's all about translation. So like, for example, we have a schema, NLM has a schema, other resource has a schema. They're distinct yet related. We both have a description. We're using schema.org, they're using something else. How do we, that's how the portal works. How do we translate that? So in the model perspective, it's like, how do you build translation across existing structures? That's option one. And then option two is start over and make a huge investment, but that's not ever guaranteed or straightforward. So to me, it's like, how do we, and some things are just not going to connect. And but trying to figure out how to translate that up front or now, is a good exercise. And I think that goes back to the interoperability push that NIH is coming with too. Like to me, that's like technically like, how do we translate across these things to get to interoperability?
I'm just thinking of what happened in industry with digital twins. They're highly standardized. And the way it happened there is it was driven by large companies. And they said, if we're going to use your component models for our digital twins, they have to be written in this language and satisfy these specifications. Otherwise, we won't care. And that's, of course, very different in this space. We don't have sort of centralized customers like that.
You've kind of got two levels of complexity. One, it's biological, and translation to biology, it's different than like an airplane wing. And two, the federal government is, and the NIH is different, right? It's not a top down. I mean, it is at some level, but we've sort of sprouted these different activities, right? So we kind of have all these so then those are the questions that we face on a daily basis. Do you say, okay, like who is, and that requires, thinking about my words here, that requires leadership, right? That requires somebody to make that decision, to say, these are the specs, this is how we're going to build it. The reality of the matter is that what you have is a lot of really good existing efforts, and then trying to layer like an idea of like digital twins on there. And so I think towards a digital twin initiative for human health, where you're based, it's almost like an ARPA-H situation. I think I saw Gary on at the Circle. He's in the video of the Circle Proposers Day. That's a huge investment for essentially a biomedical digital twin. I mean, Circle, as far as I can tell, that's a huge investment. That's a little different than kind of like bespoke, kind of piecing together existing things. So it's sort of two approaches, I guess. Yeah.
Okay, well, thank you, Reid. Thank you for the presentation today, and I appreciate it very much.
Thank you for all you're doing. I guess I should, let me just answer the IMAG question. IMAG remains, like, we do meet still. I think with the government kind of like the transition of new leadership and everything, like we kind of had sort of a pause on communications last year, and it sort of struggled to kind of get back up to speed. So it's hard to have like the regular meetings, but I have worked with Grace. We do have like monthly like IMAG meetings, not the MSM, but the IMAG. Starting up in April, and she remains very committed to it, and I think it's one of those things where it's a labor of love. And so if there's, I don't be shy to reach out and make suggestions of, like, I think we were also struggling from like, to what end, like we're asking you all for free work basically, right? So like, like what is the best solution here for how to continue these conversations? And we're open to any of those suggestions.
Okay, thank you, everybody. Thank you, Reid, and we will see people in a few weeks. Again, appreciate you for coming today.